So, we're really trying to make an effort here to engage postdocs and grad students with cutting edge research. So we would like you guys to participate to the greatest extent. And, the structure of this seminar series there'll be a research talk for the first part. And then after that, we'll sort of have a more informal discussion and maybe a broader discussion with Janne, where he can share his perspectives about science. Okay, so it's a great pleasure to have Janne here. He's the William R. Keenan Professor of Physics at Brandeis. So Janne did condensed matter physics PhD at Cornell. We completed that in 1995. And then he moved on from there to Brown, and then was a member at the IAS, where I believe he worked in theoretical high energy physics. Correct me if I'm wrong about that. Okay, and and then he moved on to become a professor at Brandeis in 1999, and somewhere during that process, he moved over to doing theoretical biological physics, and his group has done great work on chromosome organization, gene regulation, and cytoskeletal dynamics. So, in addition to being a really prolific scholar, Janne has been honored many times for his contributions as an educator. I think probably everybody in this talk will be familiar with the book that he co-authored with Rob Phillips and Julie Theriault and Hernan Garcia, Physical Biology of the Cell. I can say personally that was one of my first experiences and entrances into biology. And Janne has also been deeply involved with courses at the Marine Biological Laboratory. So I was lucky enough to participate in a course on physiology in 2016 that Janne was an instructor in. So I think Janne has touched many of our lives educationally in many ways, and so I'm very happy to have him here. So without any further ado, Janne.
All right, yeah, so I just want to thank Karna and Simon and Mata for inviting me. I was just telling them earlier, I think it's a great thing that they're doing here. I think we need more of these kinds of seminars that are, really geared towards students and less towards other faculty. So I think it's a fantastic idea. And as I was saying, what's really also awesome about these kinds of things is rarely will anyone say no if they get invited by a student. And then we've got Zoom now, which, yeah, with all its problems, gives us the opportunity to do this and not have to get on a plane and suffer through hours of sitting on a flight. So okay, so I'll start my my talk. So what I wanted to say was, feel free to interrupt me. I guess I can set the rules if the organizers don't mind. But in the sense that I'm completely comfortable, and people should just feel free to unmute and ask questions. But if the organizers want to do it somehow in some other way, I'm happy with any scheme. But I am hoping that, to the extent that Zoom allows us, we can have a little bit of a dialogue. And certainly after kind of the talk, I'll be happy to stay as long as people want to chat. So the thing I want to tell you about is this problem I've been sort of consumed with, I don't know, for about a decade now, which is the question of how cells measure length. And just to kind of get us going, thinking about this, I just want to sort of remind everyone that the sort of inside of a cell is a total mess, right? There's a hugely dense packing of all the sort of molecular constituents that this video from Ex Vivo nicely describes. Brownian motion sort of is the name of the game, and yet, right, through even though we have all this sort of randomness at the scale that you're seeing here, which is about the scale of about tens of nanometers, at the larger scales of hundreds of nanometers to microns, the life of a cell can also be quite orderly, right? And And for example, even though all this insanity is happening at this tens of nanometer scale, at the 10 micron scale, which is the length of these flagella that you see in this swimming algae, Chlamydomonas reinhardti, it is very, very regular. It does this. The algae does this beautiful breaststroke, and it does that through sort of a mechanism that's well understood, that has to do with motor proteins, which I'll talk about later, dyneins moving along microtubule filaments, which are the sort of the structural units of these long flagella, and I will get into that later. And not only do you get these beautiful patterns emerging at the cellular scale of tens of microns, but also these patterns, right, are functional, and that's sort of in a big way the idea that there is function underlying these patterns, or that there's function that's a result of these patterns, is sort of central to biology. And for someone like me, as Karma said, who really grew up as a condensed matter theorist, the idea of function is something very unusual, very new, and very exciting. One of the reasons I really have been enjoying this life in theoretical biology for the last 2 decades or so. And it's functional. And in fact, if you look at these are some measurements done by Wallace Marshall, and if you measure the speed of these single cell organisms, and the speed here you can see is at its highest about 100 microns per second, and then there's a variation of the speed. And the way they do this experiment is they create a bunch of different mutant cells, and these mutants have different lengths of flagella. So what's plotted on the X and the Y axis are the lengths L1 and L2 of the two flagella with which it's swimming. And so there's a variety of mutants, and they have different swimming speeds. And so that's how you get these contour plots. And what's shown here in white dots are the lengths of the flagella that you find in wild type cells. And what you can see is that a couple of interesting things. One is that there's the variation in the length is rather reasonably large. You can see sort of kind of a variation that goes between anywhere like 5 and 15 or so microns, but most of that variation occurs along the diagonal here. And the diagonal means that most of these wild type cells have the lengths of the two flagella equal, right? And then of course there's some deviation from the diagonal. So but those deviations which correspond to the difference in the lengths of the flagella are much smaller than the length of the flagella itself. So it seems like, and we'll talk more about that, this suggests that there's something controlling with the lengths that would like to make the lengths equal in size, and that seems to be maybe more important than having them of a very specific length. And what's also interesting is that that you see a lot of these wild type cells have flagellar lengths which correspond to the largest possible, the largest speeds that they measure, suggesting that maybe there's been some optimization in the process of evolution that produced these flagella. But of course, that's just a speculation. So that's sort of the main theme of my talk, and that is how do cells construct flagella, filamentous structures like the flagella that have a very specific length. And this question kind of arises in many different contexts. So I just showed you some data, and I'll show you more data from Wallace Marshall over at UCSF about Chlamydomonas with its two flagella. I think I'll get to and show you some of our data on structures that are very different. These are actin-based structures, unlike the flagella, which are microtubule-based structures, but they're also structures that are within the cell. And these are in yeast cells, and these are cables of actin filaments. And their role, their function is to transport material in a budding cell. And the material that's being transported is material that the bud requires in order to grow. So it serves as a highway for transport of goodies from the mother cell to the daughter cell. Actin filaments also are really important in these structures, which are observed in hair cells. These are cells in the in our own inner ears, and they have this beautiful staircase structure. So these are filaments that all come out and they protrude out of a single cell. And this staircase structure that you can see is critical to their function, which is to detect sound waves, and deflections of these filaments due to a passing sound wave actually end up leading to deflection. And that deflection actually leads to the opening of ion channels, which is the first step in the process of hearing. And then, I've been obsessed with hair cells for a very long time, but I won't be talking about them mainly because I don't have anything I think interesting to say. Another really cool single cell organism is Giardia. It's not so cool if it gets, if you end up getting one of these stuck somewhere in your intestine. And this is why you shouldn't drink water out when hiking, because they're often found in these kinds of environments. They will, they won't kill you, but you will not be a happy trooper for a few weeks, maybe months before you can kind of get rid of them. They have all sorts of interesting properties, but the thing that we've been kind of amused by and have been studying is their flagella. And unlike Chlamydomonas, they have 4 pairs of flagella, each with a specific length, and we're trying to understand how those are determined. So that's kind of the gallery of different cells that we're interested in, and so the way I thought I'd structure the talk, and I will try to be mindful of the time. And I forgot, how long do you want me to talk for? Like 30 minutes, 40 minutes, 10 minutes? I know it's more than 30, less than an hour, but I forgot it was.
I think 40 to 50 minutes is good.
40, yeah, okay. And hopefully people will interrupt me, and then I will. And that's fine, because I don't have to tell you everything I thought I could tell you. So, so as I was saying, this is our gallery. But what I wanted to really do first is give you a couple of puzzles, and those puzzles will be sort of what I will use to motivate what I will say later in the talk. I like puzzles. I've always liked puzzles. I don't know, when I was a kid growing up, I'm a little older than most of you, so I had a Rubik's cube by my bedroom, in my bedroom, and I actually used to know how to solve it and all that. So I was always motivated to do science because of puzzles. And I like math puzzles. Turns out it's just where I grew up. I think the point was that the only place I could find good math puzzles outside of mathematics itself was physics, because no one ever taught me biology in a way that presented puzzles. It was just taught as a laundry list of things. So I was really not at all interested in that. But anyway, so here's a couple of puzzles that I wanted to tell you about. One is something that happens with these algae, Chlamydomonas cells. And it's this really fascinating process whereby it's always best, I don't know, to do this by showing my arms. I think this will work. So what you see in this data, in these snapshots, and the time here is ticking away in minutes. So you can see it goes from 0 to like about 30 minutes or so. What Wallace is doing here in these experiments is at time t equals 0, he uses a laser to cut off one of the arms of this flagellum. So now you've got cell. So now you've got a cell with one arm, one flagellum. And what they observe is a process of regeneration. This process works in the following way, is that the flagellum that was left intact, its length shrinks. And while its length shrinks, the length of the one that was cut off grows. And this shrinking and growing is going on until they actually reach an equal length, a set point of equal length. And then they slowly grow together up to the original length. And you can, and so the puzzle is, how does the cell sort of achieve this, right? I've got to have my Legos here. I forgot about them, but I really want to kind of people to sort of think, kind of get a sense, and I'll talk more about this, of why this is really weird, right? Because at the end of the day, let's not forget, right? Inside these flagella is essentially, a microtubule, which is a bunch of Lego blocks stuck together. The Lego blocks are called tubulin, but who cares? They're just Lego blocks. And what these Lego blocks are doing, of course, is, they're just kind of diffusing around. Well, we'll see, it's not exactly that simple, but roughly speaking, that's what all molecules can do, is diffuse around. And on occasion, they'll bump into something that they can stick to, so they'll stick there, but then they'll fall off. And that's it, right? There really isn't anything else. At least that's what Pasteur told us. And so then how do you, for something like that, of course, you're going to keep this thing, you're going to kick this thing out of equilibrium and all sorts of fun stuff like that. But at the end of the day, how does the system of interacting, and the interactions are only short range, molecules that are a few nanometers in size, how does it know about the length of this polymer that's 10 microns in length? How does it know that how does it know that it wants to be, if I can use those words, how does it know that it wants to be 10 microns and not 1 micron, right? So that's sort of what fascinates me about this problem. This kind of, there's this bridging of scales from few nanometers, the size of an individual protein unit, to tens of microns, which is the size of the structure.
Yanni, I've got a question.
Yeah.
So those earlier data that you showed, where I think you're showing that Wallace generated mutants with different flagellar lengths. Is that, so those are equilibrium flagellar lengths somehow?
Oh, yeah. If I'm not mistaken, yeah, they're different. They're different. Most of these are, there's a bunch of genes that are known to be involved in maintenance and growth of these flagella, and they're knockdowns of different genes. And so they're mutants in that sense. And yeah, so I believe, the idea is that, so these are not experiments where it's a good question, because I also just told you about experiments where you cut the flagellum, and there there's about a 30-minute sort of period where you're not in steady state, where you have some kind of you have this interesting dynamics that I try to explain with my hands. These were presumably not done while these interesting dynamics where the lengths are changing is going on, right?
Okay. So is that to say that there's some sort of cellular control of asymmetry?
Yeah, I mean the asymmetry, because you don't see kind of these, I was talking like these fluctuations that along this axis, which would imply a difference in length. Yeah, there's something that controls the two flagella to have the same length, right?
I see. Okay.
Yeah, I mean, if you allow me to forget about the dispersion in this direction, then the fact that all the wild type cells are lining up along this diagonal tells me that there's something that's keeping the lengths equal, right?
Okay, thanks.
Yeah.
It looks like we have a question. Nusha, do you want to unmute yourself?
Yeah, thank you. I was actually wondering about the next slide, when you do this experiment where you cut off one of the flagellas. Maybe I understood it wrong, but this red line is where you cut it off, or?
No, you can see, sorry, I don't know why the red line's there as far. Okay, so I'm not 100% sure. What I am sure about is, and I don't, the data I will have a little later, is that there's the data. I didn't have it. In my grand planning scheme, I was going to show the actual data later, but since you asked, I think maybe it's a good time, and so we can be very precise. So here's the result of many such experiments, right? And the red is the one that's being cut, and you can see that around 5 minutes or so. You see how the red's up here, and then it drops down to 0. So so the laser ablation gets rid of the flagellum. What I don't know is if you cut it close to the end, whether it then shrinks and goes away very quickly, or they're actually severing the whole thing. There's different ways of getting rid of flagellum. People also use sort of shear flow to just shear it off, and you get the same kind of results. But here you can see very specific, in very detailed sense how the one that's been cut off, the red one, grows, and the blue one becomes smaller. And then you can see they reach some length that's roughly the same around this 8 micron point. And then there's a slight slope here where both of them are increasing. And if you wait long enough, a couple of hours or so, they will eventually go back to being 12 microns. And what I will tell you about is, the sort of my little theory part of the talk, even though I'm more interested in sort of conveying the excitement that I feel about these questions. But in the theory, when I discuss a little bit of theory or a little bit of simple mathematics, what I will attempt to describe is this rather fast process where the two lengths are equalized. We don't really know what's going on here. We believe that this is protein production, because when you sever the flagellum, you actually end up, if you make the estimate, you end up using quite a good fraction of all your tubulin. And so we think is going on is that there's sort of a reshuffling of building blocks between the two flagella, where the long one is losing them and the short one is gaining them. And that's fast. And then you need to make more tubulin to get them to be as long as they used to be. And so that's taking that process is happening at a longer time scale. So this is great. I'm going we're not going in order, but who cares, right? We're just having fun here. So that was the first puzzle. The second puzzle I already told you about. It's one that comes from Giardia, and there the thing that I'm fascinated by the fact is that this is single cell organism. We believe, and I should have said that, we believe that all these structures that come out of the cell, which are made of tubules, they eventually end up they're connected diffusively to the cytoplasm. So you can think of this whole cell as a little diffusive bath of particles, and yet somehow these particles, maybe the tubulin molecules, know to produce 4 different pairs of flagella of lengths which are 12 microns, 7 microns, 14 microns, and you can see the variation here in a population of wild type cells is quite small, definitely much smaller than the difference between the lengths of the flagella. And so there's something about the way that, again, there's some program, there's some network, whatever language you like to use within the cell, that tells the cell, build me 4 pairs of these flagella. And that's again, something I'm interested in. And finally, another filamentous structure, but now made of yeast, are these actin cables. And here you can see images of these actin cables, these very dark, long lines. This movie, I'm guessing it's a little hard to see. I'll tell you what you should see is there are these little white blotches. Those are actually secretory vesicles. They're just lipid sort of vesicles, and they contain a bunch of proteins that, and those lipids actually as well, are needed for this bud to grow. If you disable that transport, there's no bud growth and the cells are sick. And transport occurs along the actin cables, and these vesicles are decorated with myosin molecules that move from the minus end, which is towards the end of the cell, towards the plus end of these actin filaments, which are all at the bud neck, which is the place where the bud is shaping away from the mother cell. So those are actin cables, and the thing that is really that a postdoc that's in our group recently discovered is that if you look at yeast cells of different size, then the length of these cables scales with the length of the cell. So in other words, if you measure the cable lengths, as shown in this plot, here you have four different, sorry, in this middle plot, you see the cable lengths, and these are data from all four different yeast cells, haploid, diploid, and then a couple of different temperature-sensitive mutants that we built that make the cells grow abnormally large. And the actual lengths of these cells, the length being the distance measured from the bud neck to the end of the cell, they also vary between 4 microns and about 8 microns. So we have a variation in length of about a factor of 2, which means that we have a variation in volume of about a factor of 8. But then when you plot, just when you scale the lengths of the cables with the lengths of the cells, all these distributions basically collapse onto each other. So this to us was just really exciting, because we think it's telling us that whatever the mechanism is that is controlling the lengths of these cables, it knows about the size of the cell. It produces scale invariant cables, right? Which to us is sort of interesting. So anyway, to summarize these two puzzles or engineering challenges faced by Chlamydomonas cells and yeast cells, even though here I drew Chlamydomonas twice, one is how do you assemble out of a common pool of building blocks? These are the little green circles. Those are the tubulin molecules. How do you assemble two protrusions, two filaments of equal length, or for that matter, 4 pairs, where within a pair the lengths are equal, but each pair has its own specific length, which was the case in Giardia. And then if that's not enough for you, then how do you self-assemble protrusions or filaments such that their size properly scales with the size of the cell? And I've shown you data of this kind of scaling. I just showed you data for this kind of scaling for these actin filaments in yeast cells, but Wallace has published data actually for the same kind of scaling occurring for flagella in Chlamydomonas, which I'm actually kind of confused about, because I have to get back to him and talk about that. I don't know what happens with the swimming then, right? So anyway, but there seems to be some scaling there as well. That's still sort of unpublished, and but I'm just saying there's some suggestion that also these flagella and Chlamydomonas have a scaling property, that they scale with the size of the cell. And I just put up this video. So those are 2 puzzles, and what we have to keep in mind when we think about this is that these filaments, they're very dynamic. And what I mean by that is that their length, even though it's maintained over maybe hours, the composition of the filament is constantly changing. If you follow molecules, they're constantly coming on and off the filaments. And to give you a sense of how fast in these cables in yeast that are, as you saw, about 6 or 7 microns in length, if you add a drug to the cell that sequesters monomeric actin, so that it disables polymerization, it takes 1 minute for the whole filament to disassemble. So in 1 minute, it's gone. So that's the rate at which you're turning over the constituents of these filaments, but the structure, if you will, remains for much longer, right? So it's stable, even though all the molecules are constantly exchanging, right? And so this little video, again from ex vivo, just kind of gives you that sense that these cytoskeletal structures are constantly being assembled, and this is here you're seeing assembly of actin, and then constantly being disassembled. One of the main ways actin disassembles is through cutting or severing. This is a microtubule. Since I haven't shown you microtubules yet, it also assembles, disassembles. Outside of cells, it often disassembles in this kind of catastrophic way. Inside cells, not as often. And then, of course, I'll talk a lot about this motor transport, because I will try to sell you a theoretical idea that it is actually motor transport along these filaments that's critical to controlling the length. So that's going to be kind of a key idea. Okay, so that's so then let me tell you what's the sort of the key sort of idea that we borrow from mathematics to try to get some understanding of these observations that I've just described for you. And we just essentially think of this, we adopt sort of a way of thinking that just comes directly from dynamical systems. For us, or at least for us, it seems like the simplest possible way to describe something like a filament that grows and that is assembled and disassembled and reaches a fixed length is through a simple dynamical system with a fixed point. Nothing more complicated than that. And what do I mean by that? Well, imagine that you have a filament of length L, and the process that we're going to consider is the process of adding a unit, a subunit, a monomer to this filament. And by doing so, it'll make the filament have a length L+1. And then, of course, the subunit can also fall off, and then the length will decrease. And therefore, we can write down a very simple equation that says that the rate of length change is just the rate of assembly minus the rate of disassembly. That's just mass conservation, if you will. And then if so then within this framework, and this is the framework within which I'll be discussing all the results, this simple dynamical system, one-dimensional dynamical system. So for example, if you want this dynamical system to give you a fixed length, all you have to do, and then a lot of the rest of the talk will be trying to understand how this is actually achieved, but all you really have to do is somehow set up assembly and disassembly rates that depend on L. So you have to set up some kind of feedback where the rate at which you add monomers or the rate at which you remove monomers depends on the full length of the filament. And if you do that in this following way, where the assembly, for example, decreases with length and disassembly increases with length, then obviously there's going to be a fixed point when the two are equal. And the way I've just drawn this fixed point is stable, right? Because as soon as the length goes a little bit above L star, disassembly is greater than assembly, and you will go back to the fixed point. And similarly, if you're slightly below L star, assembly will be going at a faster rate than disassembly, and you will increase your length toward L star, right? So that's the level of mathematics that I'll be using. So I've sunk very low compared to my glorious days as a field theorist, but I'm very happy about that because I think I've discovered that what I really always want to do is simple math. Okay, so in fact, I've gone to a few conferences. I won't name where they were, but I was criticized for my math being too simple, which tickles me to. It just I don't know. I was just tickled pink about it because I don't know. I used to be criticized for my math being too complicated. Just can't please people with math, I mean. So anyway, so let me try to then convince you that, or let me try to tell you then how you can, within this dynamical systems framework, how you can build a system that regenerates and controls length. And then I'll tell you also a little story of how you can build a system that assembles filaments whose length scales with the size of the cell within which it's assembling. So that's going to be my goal, and we'll see how far we get. But also, if you have any, this would be a good time to ask a question. If you have any questions about, like, if you're lost, if I've just completely lost you, this would be a good time to say I am completely lost, because at least I want everyone to understand what the questions are, and then going forward, what the answers are. I always tell my students, who cares? I mean, the questions are the thing that are really, I always feel are the really important thing. So please ask if the questions aren't clear. Yeah.
I mean, maybe I'll have a question here, which is, so should I think about I'm in a reservoir, so I have these growing filaments, and I'm in a reservoir where I kind of have a limitless supply of monomers at a fixed concentration, or do I have some finite thing and now they're closed because of I guess if I just think of any of your systems, and I think of how many monomers make one of these cables compared to the size of the system in terms of monomers. What are those relative units?
Oh, yeah, very good. So like for a clammy cell, so the answer to your question is yes and yes. But for from a clammy cell, if I look at all the green dots that are in the filament, that's about 40% of all the green dots. So it's a sizable share. In Giardia, we don't have a good sense of how many tubulins there are, but given what we know, we think the amount that's in the filaments is actually a very small, less than 10% fraction of all the tubulins. So in that case, we actually think it is more of an infinite reservoir. With actin, we think it's again a fairly sizable amount that's in these filaments, but things get a little complicated there, because at the end of the day, of course, what matters for these rates of monomers coming on and off is the free concentration, how many monomers I have diffusing around in the cytoplasm. And at least for tubulin, there's some really beautiful papers recently from, who was it? One of the older Harvard dudes. I forget, Mitchison, I think. Anyway, so they have some really interesting results that show that there's an interesting feedback at the level of gene regulation for tubulin that seems to be responsible for controlling the free concentration. So which suggests that you start building these structures, you start depleting tubulin, but then something else kicks in to keep the free concentration. And again, free concentration is just a key word for saying something that will keep the rates that depend on that concentration constant. So yeah, there's all these things going on, Madhav, and it's interesting to think about all of them. And we do think about all of them, and you'll see in some cases we'll consider the pool to be limiting, but actually in this talk I'll mostly consider that this free concentration that sets the rates, that is actually constant pretty much. But well, maybe I'll change my mind at some point. Yeah, we'll see. But all those things happen. I want to emphasize, and it's very interesting. People are just starting to kind of, there's more and more papers now where people are starting to think really hard and quantitatively about like the simplest questions you can ask. If a cell gets bigger, does the concentration of the relevant proteins you're interested in that determine the physiology, is that concentration staying fixed or is the number staying fixed? And there's some really nice results from Jan Schottheim at Stanford showing that there's actually some genes that produce proteins at the constant concentration, and it looks like it's at the level of transcriptional control, and some that keep it at constant number. And then it looks like maybe some that are in constant number have something to do with determining cell size, because of course, if you increase cell size, you start diluting the ones that are constant number. All sorts of fascinating things. The beautiful stories also coming out of Dresden on spindle assembly, and also having to do with exactly the questions you're asking, like how are the tubulin molecules partitioned? And actually, in that case, it has to do with some kinase that phosphorylates them. Anyway, so yeah. Sorry, I went on some crazy tirade that meant probably nothing to anyone except my mind. Yes, there was a hand up. Go ahead.
Yes, so I was wondering if the rest of the talk is going to focus on this, you can completely ignore the question.
Yeah.
But mathematically, it was simple what you described, but physically, what kind of mechanism causes the rates to go that way of the assembly and disassembly?
That's going to be my next 3 slides. Thank you.
Okay, then we completely, you can just go over the question.
No. I'll explain that next. Yes. So if there are no other questions.
Can I ask a related question?
Yeah, absolutely.
I'm trying to understand why they are like go to equal length. Is it that when they disassemble, is it more likely that the other one is going to take them always if the supply is limited? If it only like comes off, if it's going to go to the next one?
Yeah, you can actually show that just having a limited supply will not do that. And we can, if you want to hang out after the talk, we can actually, I can pull out my iPad and we can sketch that out on the iPad virtual blackboard. But So we thought about this. This is a really interesting question. To what extent can we, to what extent just having a limiting pool will produce this kinds of behaviors, and at least the simplest version, which without any bells and whistles, doesn't do it. Took us a while to figure that out, actually. So I'm not saying to me anyway, it wasn't an obvious result, but good question, yeah. Okay, so just showing the data again, we're all now familiar with the fact that you cut off one, they regenerate in length. And like I said, I'm going to focus on this initial phase where there's an equalization of length, right? So that requires, I think it's clear to everyone that it requires a little more than just this little diagram, right? Because this little diagram just says if you build this, if you have some kind of feedback, you'll get a fixed length, right? So the regeneration process is somewhat different than that. So then how do you even get length dependent assembly and disassembly? And so what I'm going to argue for most of the rest of the time, and I'm going to stop at 2:30, I guess, regardless of where we get, is I'm going to argue that it's all about motor transport. And what do I mean? Well, if you take this chlamydomonas cell and this is an experiment out of Ahmed Yıldız's lab at Berkeley, did some beautiful experiments, this paper in eLife. So the cell is in the middle. You don't see it because it's a fluorescent microscopy that they're doing here. The two flagella have been splayed out. And what you can see here are little white dots going up and down. And these little white dots going up and down are fluorescently labeled kinesin molecules. And these are fluorescently labeled molecules that are being transported by kinesin and dynein. Because for microtubules, what goes on is that kinesin molecules will transport their cargo in one direction. And in this particular case, the way that the microtubules are oriented, the transport by kinesin goes from the anterior, from the cell body, to the posterior, which is the tip of the flagellum. So up here, the tip is this tip here of the flagellum, and the cell is here, and the beginning of the flagellum is roughly here, which would be corresponding to that point on the top on this image. And you have actually a very elaborate, process that's been studied a lot by molecular biologists and cell biologists of something called interflagellar transport, which is transport of proteins by kinesins that move along microtubules. Once they get to the end of the flagellum is this whole thing inside their microtubules that we were talking about. Once they get there, this whole complex of proteins falls apart, and then through some magic of well, not magic, but just things are again diffusing around, bumping into each other, and the conditions are good enough or right in terms of their concentration, so you'll get another complex assembling, but this time with a dynein molecule that manages to take all these things back to the cell body. So that's interflagellar transport. And what's critical to our problem of growth is to note something that people have known for many years, which is that when the filament is growing and disassembling, all the growth and disassembly actually happens here at the tip, at the plus end. So the tubules, the tubulin, which is the green spheres here, they're being added to the filaments and they're falling off the filaments at the tip. And the molecules themselves, the building blocks, are delivered to the tip by the motors. So they don't get in there by free diffusion, they're delivered by motors. They also, of course, get in by diffusion, but there's motor transport. And so the key thing to this is going to be super important, is that all the assembly, not so much this delivery, we can kind of ignore about that, but the key thing that's going to be important is that all the assembly and disassembly happens at the tip, okay? And so that's what I wanted to, that's idea number one, and we're gonna have a couple of, or fact number one, and we're gonna use a couple of facts to construct sort of this feedback. Fact number two, and this was critical, and this changed everything, at least for us in my group, is the observation that Ahmed and his group made about transport, and what they discovered. And without going into these images in detail, I'll just tell you the result. What they discovered is, as shown in these red and blue images, the red and blue lines, these are just, let's focus on the red line, is that then when they fluorescently label one particular protein, it happens to be the kinesin, but it doesn't matter, for us, it won't matter. They see that the molecule moves from the cell body to the tip as it should. If it's a motor, it just follows x equals vt trajectory, so it moves at a constant speed, so you get just this nice straight line. And then when it gets to the tip, you see it kind of stalls. All that we knew as well. What's happening is that when these motors bring the cargo to the tip, the whole thing falls apart. But then instead of that's actually the blue line, sorry, because they're two different molecules. So the blue line is the kinesin, sorry. So the blue line, you see, it stays there, and then after it does actually a random walk. It's diffusing. It's not being taken back by the transport in the retrograde direction. The red molecule is, and the difference is they're just two different proteins. So the blue protein, if you will, and it's blue in this picture, the blue protein gets directly transported to the tip. Once it falls off the microtubule, it does not go back on the train. It diffuses back out. The red molecule goes on the train to the tip, falls apart, and then catches the train back to the cell. And that's, you can see the red traces again are following straight lines with a speed that's roughly the same. The kinesin and the dynein have roughly the same speed of movement along these filaments, and the speed is microns per second essentially. So you zip up to the top of this flagellum in about 10 seconds. Okay, and that's fact number 2. Some molecules on these trains there are molecules on these trains, proteins, that are directly transported to the tip and then released so as to diffuse. And that's going to be sort of what we have proposed as the key ingredient to this whole business. Because, and you can convince yourself that of that, and I'm going to show you a slide with flash a little bit of essentially, physics, 101 type analysis of diffusion. But if you have such a process where you have molecules transported by a motor to the tip and then released, then that process will create a gradient of those molecules. In fact, it will create very simply a linear gradient. And you can show equally if you're bringing red molecules to the tip, releasing them, but then they can't get on the train that's going back, but they can hop on the train going to the tip at any position in the filament. Then you again get a gradient, but it's an exponential gradient. So this linear gradient you get if the red molecules, as the ones that are observed by Yildiz, if they get released at the tip and then can't jump on the train until they're actually, they can only jump on the train if they're coming in from the cytoplasm. So the ones that he tracked exactly do that, and he observed, I should have had that I should have had that data plot. They observe a linear gradient of these molecules, as you should from just diffusion, because what's really happening here is in steady state, the flux of these molecules coming in, call it R, has to be balanced by the diffusive flux of these molecules coming out of the flagellum. And that's just here, dc dx times the diffusion constant, A being the cross-sectional area. So if you just solve that, and if you have a constant flux of molecules into the flagellum, and you solve, you get a nice linear gradient, which is what they observe. But with a linear gradient, now it turns out it solves all the problems, or both controls length and leads to regeneration, because now you can write down the dynamical equation as in more detail. So for example, if you just take, and we've measured this in Giardia, there's some issues. whether the rate of injection in clammy is constant or not. And for the aficionados, it's not, and that complicates things, but actually ends up being, in my opinion, a bit of a spandrel when it comes to thinking about length regulation. So I'll just say a little white lie here and say that for clammy, let's assume that the rate of microtubule assembly here is fixed, and I mean that it's length independent. It's not, but let's just assume it is, because the other thing just makes the algebra more complicated and doesn't lead to anything interesting. And so if you have how is this the rate of microtubule assembly? R is the rate at which the microtubules are injected in, and then it's just going to be proportional. Well, I just wrote it here. It's proportional to the amount of tubulin, that just to deal with the fact that there is a limited amount, but again, not so interesting. So there's a constant rate of microtubule assembly simply by the fact that there's a constant rate of injection of these microtubule particles into the flagellum. So the microtubule particles are the tubulins, the green guys. So they're just they're brought here at a constant speed and a constant rate, and they're going to be incorporated in the tip. So there's some constant rate at which they're incorporated, right? So that's the simple thing for assembly. Now for disassembly, now I'm going to hypothesize this. This has not been proven for flagella in clammy, but we have done measurements and shown that this actually is the case for flagella in Giardia. So I'm going to assume that there is a protein, and we think it's kinesin 13. We think we know what the protein is, but anyway, that there's a protein that's riding on these. That's the yellow one that's shown here. There's a protein, the yellow protein, that's riding on the motors towards the tip, and then it's released, and then it diffuses back, just like the one protein that Yildiz saw doing that. So we're going to assume that one of the proteins that does this kind of motion, that's a combination of directed motion towards the tip and diffusion back, is a molecule that assists in depolymerization. So there's many proteins that will bind to the ends of both actin filaments and microtubules and induce depolymerization. Kinesin 13 is probably the best studied one. It's a non-processive kinesin. It just happens to share homology with the motile kinesins. So okay, so then what happens then? Well, if you take my two things that I've assumed. Well, first of all, you can work out the theory. It's very simple. Because you have a linear gradient, what happens is as the flagellum gets longer, there's a larger and larger concentration of these yellow molecules at the tip. So if the yellow molecules, again, are injected here at a constant rate, they travel at a constant speed, they're released and then diffuse, you can easily convince yourself that because what's really fixed by the rate and the other parameters is the slope of the gradient. As this flagellum gets longer and longer, the concentration at the tip gets bigger and bigger. But if the concentration corresponds to molecules that depolymerize the flagellum, then of course what you will get is a depolymerization shown in this figure below, a disassembly that grows with length. It grows linearly because the gradient is linear. In the presence of constant assembly, you will get, of course, a fixed point, as I discussed before. So that's the idea. There's a few moving parts, but I hope it's kind of clear. So assembly is just proteins are delivered to the tip. Disassembly is due to these yellow molecules you see in these pictures, and their property is that they're brought to the tip by motors and then released, and then they diffuse. And if you do that, you will set up a situation where the concentration at the tip is length dependent, and therefore any kind of chemical process that involves those yellow molecules will be proportional to the length. In particular, we assume here that process to be disassembly. And moreover, that will simply also give you equalization of lengths, because if you just write down now the equations for L1 and L2, of course, what's going to happen if the two lengths are dissimilar, the flagellum that has a shorter length will have fewer depolymerizers at the tip, so it'll grow. This one will have more, so it'll shrink. and they will equalize. And you can just see that easily by just writing out the equations and showing that the rate of change of the difference is actually exponentially suppressed. Goes exponentially to 0. And that's what we see. We see this kind of fast exponential equalization of length. So this little mechanism explains both that you have a fixed length and the fact that you equalize lengths. And if the math doesn't convince you, then this is a student of actually Ariel Amir's over at Harvard. He did a nice little simulation, and all he simulated was literally a reservoir of motors, yellow proteins, which disassemble, and green proteins, which are the tubulins. And the thing that happens is the red motors, when they attach and go up to microtubules, they can carry with them the green and the yellow guys, and the yellow guys diffuse back. And if you do that, and you start a simulation with two flagella of the same length, which you see here on the right, and you cut one, which we'll see in a second, so the bottom one will disappear. Here they're just at the same length. You sever, and the top one disappeared, and you quickly get this equalization to a smaller length. And no magic required, just this kind of coupled transport and diffusion. So I should probably stop there because it's 2:30. Maybe I'll finish in the following way. First of all, it turns out this is not just a pure figment of our imagination, even though initially we just hypothesized the existence of these molecules. Then we paired up with this wonderful grad student who's now a postdoc with me, Shane McAnally, and he actually searched for precisely a depolymerizing protein in these Giardia cells, because he was working on Giardia and not Chlamydomonas, and the black spots are these kinesin 13s, and you see exponential gradients of kinesin 13, and you see that the gradients are such that the longer flagella, remember in Giardia you had flagella that had longer lengths and shorter lengths. The flagella that are very long have a much bigger concentration of kinesin 13 at the tip than the cells than the shorter flagella. And so, the thing I really like about this mechanism, like one of the things, that's been really sort of a fascinating discovery in cell biology, right, lately, and that everyone's heard of, I'm sure, is this kind of idea of liquid-liquid phase separation. And that's just, one way to create compartments within a cell which have concentrations of molecules that are very different than the rest of the cytoplasm. Another way is the one that we've known for hundreds of years, which is just put a membrane around, right? And so what I'd like maybe to go away with is the idea that in these protrusions, the tip actually acts as it's basically diffusively uncoupled, in the sense that even though you have one concentration of the molecules, those yellow molecules in the bulk, you can get any concentration you like at the tip, even though everything is diffusively coupled, and you can tune that concentration at the tip by changing the length of the flagellum. So the tip of this flagellum acts as a sort of a compartment with a concentration that's very different than one you find in the cell body. So that allows for this magic to happen. And like I said, we've not done experiments in Chlamydomonas, but in Giardia we see these molecules really having this kind of gradient. And I didn't get to talk about yeast. That's fine. So I will not tell you about yeast, but what I thought I'd finish by saying is that both the yeast case and this case, we think kind of are the case of scaling in yeast and the case here actually end up being both described by the same idea, and without, the same idea, meaning that you can set up a gradient of proteins as long as you have polar transport of these proteins. In the flagellum, the polar transport is very well known. It's by this interflagellar transport that takes proteins from the cell body to the tip of the flagellum, and that sets up a nice exponential gradient. But what hasn't been observed yet, and we're trying to see this because I'm convinced it's there for a variety of reasons, is that we believe that there are similar kinds of gradients in cells like yeast. There you have a cortical collection of actin filaments. They're along the surface of this cell, which is more like an ellipsoid. And they too provide tracks. These filaments provide tracks for transport. And if you have molecules that diffuse within the cell and then are transported to the pole and released, if you just do the math, and the math corresponds to, like, because you're solving a diffusion equation in steady state, it ends up being electrostatics and using image charges and these kinds of things that we all learn in at some point in our teaching, in our schooling as physicists. It turns out you can create gradients that are scale invariant, which is something that's actually kind of interesting in general. So if you have a bunch of red molecules, I'm saying in a sphere, and these molecules, all they do is diffuse, but when they get to the surface of the sphere, they get transported to the pole and released. Then if you just do, this is just simulations to show the point, if you measure the concentration for different radii of the sphere, they look very different. But if you scale the distance from this pole by the radius of the sphere, all these curves collapse onto each other. And it's a simple exercise in solving the Laplace equation in a sphere to convince yourself of this. So I will stop there. Okay, kind of one of the key questions that I try to address is, what is the nature? There was the question that was actually posed early on. What is the molecular nature of the feedback? So I hope I've given you some ideas of that. In yeast now, what we're finding is this feedback seems to be very robust to knocking out individual genes in the network. And we're very interested in that because it seems like, actually, at least in yeast, this feedback that's required to make these cables of a fixed length is, in some sense, which I don't really understand or have any idea about, is in some sense more of a network property than an individual molecule property. And there are other examples in cell biology where this is the case, and I'm happy to sort of wildly speculate with you in a second about that. But I'm excited about that. I'm excited about, the idea of, networks which have functions. I'm excited about ideas about what are good mathematical frameworks beyond this kind of dynamical systems, which is limited. And so happy to kind of vex poetic about that with you after I just tell you who the key actors in this drama are. There's a lot of people who have sort of contributed to these ideas. A lot of friends of mine, a lot of Ariel at Harvard's been very fun to hang and talk to about this for a couple of years now. Bruce Good is a cell biologist at Brandeis who I've been working on actin cables with for the last 5 or 6 years. These are some of my graduate students who've developed most of the mathematics of this, and Aldrich is a very brave graduate student who is a physics student who's actually now trying to measure these gradients in yeast and is getting his hands wet and dirty in Bruce's lab. But I'd really like to point out Shane, who's absolutely the hero of the story, because a lot of this was inspired by his experiments when he was a graduate student and on Giardia, and now he's doing some really phenomenal work at Brandeis on these yeast cables as well. And it's allowing us to think really about these mathematical ideas, because Shane's just been incredibly good at getting us, providing data which, prior to him working on this, we thought would be impossible to get. So I think I will stop here and stop sharing, and you guys, what you'd like me to do next.
Thank you so much, Janne. That was a beautiful talk. I think there's one question about the talk in the chat, so I'll just read that out loud. So the question is, the mechanism you propose can explain the fixed length, but how about the initial refraction of the intact flagella after cut? How are the
retraction? Sorry, retraction. Yeah.
Yes, sorry, my mistake. How do the two flagellas communicate through the shared pool of motors?
Yeah. Oh boy. No, it's a great question, and oops, I better share my screen. So I think the short answer is, darn, I don't think I have a good, simple, and if I did have a good simple idea, I've forgotten it by now. Yeah. So the retraction is puzzling. All I can say is, I guess, you know,
we might have lost him.
Uh-oh. He was the main guy.
Okay, somebody entertain us for a few minutes.
That's the kind of thing that doesn't happen in person.
At Hogwarts, it does. They just apparate.
Send Yanni a message.
He'll be back, I'm sure.
Yeah.
Sorry, something happened.
Wrong.
I think what happened was me stupidly clicked a leave. I don't know why. Can you just let me share again? Sorry.
Yeah.
Sorry about that.
No worries. Okay.
Yeah. So let's see if I can answer the question in a coherent way. I guess, yeah, maybe this isn't satisfying. So let's see. So in this simulation, because the question was, what is the mechanism of coupling? And it's absolutely that everything is shared. So the motors are shared, the tubulin is shared, and the depolymerizers are shared. And the rules of the game is the assembly in these complexes only happens at the tip, at the base of the flagellum, and then there's motor transport and release of both the green and both the tubulin and the depolymerizers at the tip. And the tubulin gets incorporated, and the depolymerizers, they can bind to the ends, depolymerize, but then their dynamics is just diffusive once they're at the tip, right? So that was the idea. And so, you know, I mean, what this does set up is that, right, so if you cut a flagellum, right, then, you know, the point is, you know, the rate at which you're assembling these structures here at the base, that rate is just dependent on the concentration of all these molecules in the cell, right? Because these guys in the cell, they're just going to diffuse around. Eventually, they're going to hit here and do some chemistry magic and assemble, right? So if this guy is very short, the rate at which you're throwing the molecules up here is going to be the same as here, right? And what's going to happen is these guys are going to take up more, because there's no it's just going to take them up and start growing because there's no disassembly. And then the reason you'll get this guy disassembling is because you're depleting molecules here. So as you deplete molecules here, the rate at which you're injecting here is not going to keep up with the rate of disassembly up here. So this guy's going to start disassembling. And you know, I think that's a word story that works, but I forget exactly how it works out in the math. I just have to. This was actually a subject of the paper we wrote with Ariel that I'm forgetting exactly how it all worked out. But anyway, sorry, that wasn't very satisfying. It wasn't very, I think, very clear, because it's just kind of confusing my head right now. So I apologize.
Just so I understand the phenomenon. So if you cut a flagellum in half, what you see is that the remaining stock will retract into the cell, and then they'll equilibrate again to equal length. Is that right?
Right.
Okay.
So at least and that's, you know, this is like, so this simulation here is literally just what I said, just a bunch of red, yellow, green molecules. Because at some level we also, we have some math that goes with this, but it gets a little. That's why I don't have a simple explanation. I don't think I've ever gotten to a point where I understood it in some very simple way. I mean, if you write everything out, it works. What I'm trying to say, if you write out the full equations for the transport and diffusion, etc., you can just compute it. I wish I had a better. I don't have a very simple understanding of it, but. So what's going on in the simulation is everything's being changed. And at this point, at 100 minutes, what Kartik did is he just removed one of the flagella, the blue one, and then and you can see that on the right-hand side. And then what happens is the red one goes down. You can see it going down. The blue one goes up, and then once they equalize, they grow together because here. At this point, I mean, as soon as you cut, he starts also producing more tubulin in the cell. So this was trying to simulate the whole process. But the key thing that I was focusing on was not this growth that happens after equalization, but the equalization itself, right? And at least, kind of a simple way that you can see that there's a force that leads to equalization is if you just write out the equations for the two flagella, right? So what does this say? It says that the rate of change of the length of one flagella of either, it's the same equation, just there's a constant rate of assembly and then a rate of disassembly that's proportional to the length. And the reason it's proportional to the length is because the concentration up here in the tip is proportional to the length. And so if you just take the difference in these two, you'll see that the rate of change of the difference in length is proportional to the length difference with this negative coefficient. So it tells you that if you set up an initial condition where the length difference is not zero, this exponentially goes to zero. And that's exactly what you see in this experiment, that this difference in length, which would be the distance between the blue and the red, that goes exponentially to zero.
Right. I see.
So yeah, that's sort of the idea.
Looks like Madhav has a question.
Yeah, go ahead, Madhav.
Yeah, Janne, thank you. Elegant as always and inspiring. I wanted to ask you that, this linear versus exponential, some systems are linear, some systems might be exponential. Am I right that if I could, again, it could just be that the measurements are not possible, but if I could look at the jitter in the length, so let's say I'm at the fixed point, and now I just look at the fluctuations in the length around the fixed point. I was thinking, does Janne really care about whether it's exponential or linear? Do you really care? Because both of them will have a fixed point that in principle, some parameters will modulate. Do you care? And one thing that jumped out was as soon as you think about it statistically, the fluctuations in the exponential case will be far more severe than in the linear case. If you're somewhere near the heel or something like that. So but then that made me realize that really a way of empirically discovering whether you're in the linear or exponential regime, look at the fluctuations around different set points. And that would kind of tell you whether you have a linear or exponential. I wonder what you thought of that.
We're finishing the paper on what you just said.
Okay.
But actually, even before, we're it's actually a beautiful system for studying fluctuations because these things you can see in a light microscope, and the fluctuations are large. You can see it in that plot that I got. So if they're easily measured, easily.
So that wasn't measurement noise. That's not measurement noise.
No. So Wallace, right? So Wallace, and this is one of the kind of benefits, if I could say, of being on Zoom and all this insanity we've been dealing for the 2 years was Wallace and I paired up because he put an archive. He put a paper up showing fluctuations, and he did a really wonderful job showing that the measured fluctuations are way above noise. And then we proceeded to do exactly the kind of analysis you suggested, and we learned some things that actually we couldn't learn about exponential versus linear, but we learned some other things about the molecular mechanism, about the details of the molecular mechanism from the fluctuations, as you might expect, because you're rightfully saying, if I have this fixed point, right, And basically what I've constructed is an Ornstein-Uhlenbeck process. If I sit exactly at the, if I sit at L star, the fluctuations around it are going to be the fluctuations of a harmonic oscillator due to noise, which is not thermal, but is due to all this chemistry that's going on, right? And then some aspects of the chemistry you're going to be able to read out from the size of the noise. And I can tell you, like, the thing that we read out, so to speak, was actually a big surprise to Wallace, who's been working on this for 50 years, which I think, to me, is one of maybe 2 or 3 accomplishments I've had in biology. And those always have come, for me anyway, in the same way, where from the math, I was able to show something to my biology friends that they found surprising. Because, often, or most of the times, I would say I show them stuff that they find completely obvious. And that's fine, because, I'm not deterred by that, because that's just how I understand stuff. And if I'm going to think about biological systems, I better be writing some math, or it just makes no sense to me at the end of the day. But in this case, he was surprised, because what we found is that the noise was such that it suggested that when, we don't know which one it is, we suspect it's the disassembly process, but when the elementary step, so to speak, of the assembly or the disassembly process cannot involve the single molecule, but in fact has to involve a huge chunk of the filament. So there's something going on, which we don't understand, that's making this process much more noisy than you would get if you assumed that the units that were added and subtracted were single monomers. Okay, and the words you can use, which are, and I think the math is somewhat similar, is bursts of assembly and disassembly, and these are the kinds of words that people have used when they've looked at noise in gene expression, because they're, again, pioneered by Ito Golding's experiments in 2005. When you look at RNA production from a gene, and or certain genes, not all genes, what you find is, no production for long periods of time, and then periods of time where you have basically constant production. And so you produce RNA in bursts, as opposed, it's not described by a simple Poisson process. So I think or you can think of it as a Poisson process that's adding and removing large numbers of units in a given time interval. So that's what we're finding for these filaments, and that was not what was, at least by someone like Wallace, expected, and sorry. And what's really fascinating, right, is having 2 filaments, and now you can study fluctuations of this one and this one, and you can ask, how are the fluctuations correlated? For example, if they're sharing a pool of monomers, meaning that, if one grows, I've just convinced you that one grows shorter and the other one, one grows longer and the other one shortens because there's molecules being siphoned off. That would imply that the fluctuations can be anti-correlated if you work through that. So Wallace measures the fluctuations and they're positively correlated. We are completely clueless about why, but it's exciting. We have no idea. Oh, and by the way, we've also looked in Giardia, which is, separated from clammy as much as we are from yeast or something, a few 100 million years. We see the same size fluctuations and we see the same correlations versus anti-correlations, even though many of the molecular details are totally different. Some of the proteins are not even present in Giardia that they are in clammy. And so what Shane wants to do, and I'm hugely excited about this, is actually take a bunch of protozoa, a bunch of stuff that has flagella, and they're from all sorts of branches of the tree of life, and start actually really measuring these kinds of things all across, and see, what's universal, what's not. And I don't know what we'll learn, but it just sounds like a lot of fun. We'll see.
Because as you said, you can just do this in a light microscope.
Yeah, Yeah, you just need to be able to grow these things under a microscope and keep them in your for a long, for a while. So they don't swim away. Some are, that kind of stuff, but people, that's not that hard. Yeah. But you just need, it's nothing fancy, just a light microscope, because the fluctuations, like lengths are tens of microns and fluctuations are a few microns, super easy to measure. You're not going to bleach anything out. You can measure these fluctuations till the cows go home, right? Or until the cell divides. Yeah. And actually, that's also interesting what happens during division. And so there's lots of kind of interesting other questions.
Beautiful.
Yeah, amazing. Why don't we, I think we have a little bit of time left, and Janne, you should let us know if you need to go, because
no, I'm good. I'm happy to chat. I did have some meeting, I think at 3:30, but otherwise.
Okay, maybe we can take a little bit of time right now for people to ask maybe more broad questions. And I'm going to stop the live stream right now. So this terminates.